I'm Alexandra Zidovska from Center for Soft Matter Research, Department of Physics at New York University. Thank you so much for having me here today. I would like to thank the organizers, Mike, for organizing this fantastic meeting. I very much look forward to listen to all the exciting latest stuff. So what I'll show you today, somehow it keeps coming. Okay, thanks. What I will show you today, you saw my talk was listed at a more general level as the biophysics of the human genome. I have specified it a little bit more. For this morning, I will tell you specifically a story. What have we learned about the interface chromatin? So the genome during the interface, so during the time between two cell divisions, specifically how we have identified there a salt-gel transition that occurs upon cell differentiation. So I will walk you through all the details, and you will see here these magenta little regions. Those will be the little islands of gel phase that will emerge. All right. Apparently not. Archive doesn't work. This is okay. Now it does. So let me start first by introducing my system, since I imagine I'm the first one here speaking about the genome today and at this conference since yesterday. So the genetic material in higher organisms is stored within the cell nucleus. As you all know, the genetic information is encoded in a long DNA molecule. And what is so tantalizing about it is that as humans, we have 2 meters of DNA in every single cell nucleus in our body, and the cell nucleus is barely 10 microns in diameter. So you see, and here the cell is at the order of tens of microns. The nuclei here are highlighted in blue color. So you see, it's a true engineering feat how the nature actually manages to pack 2 meters inside of a 10 micron nucleus. So of course, the DNA is actually not naked inside the cell nucleus. However, it is complex with proteins called histones. It forms these protein particles that we refer to as nucleosomes, but that is here unnecessary detail. What I want to draw your attention to is that we have the DNA wrapped around these protein particles forming these pearl chains. And these pearl chains, this we refer to as chromatin fibers. So those are these spaghettis here inside of my cell nucleus at this cartoon. You all remember from high school biology that as humans, we have 23 pairs of chromosomes. So what does it mean within this picture? That means that you have 46 polymers, so 46 of these spaghettis inside of the nucleus. Now, I'm bringing here the analogy of spaghettis intentionally, since I want you now envision that you're cooking 46 spaghettis at home and now color each of them by a different color in your mind and imagine it. Okay, so this is what I anticipate you have envisioned, right? So this is a polymer mixture in thermodynamic equilibrium. This is what physicists we somewhat expect, that it will be a mixed mess. But this is in contrast what you actually find in the cell nucleus, where these spaghettis actually sit in well-defined regions that we call chromosome territories. So this is your first sign that we are here working with a system that is not in thermodynamic equilibrium, and you'll see, even coming later, that it has many additional cues for us. So, but first point or message that I would like to hear to transmit is that the genome clearly is organized. Indeed, it's organized at hierarchical level across different length scales, and that in very particular detail. So we have already discussed the small length scales organization having the DNA wrapped around the nucleosomes. Then this fiber forms loops. These loops make further more complex structure that we refer to as topologically associating domains. These can then further form clusters and compartments. And finally, the whole polymer itself, one chromosome, forms the territory that are here colored by each respective color corresponds to one territory. So one thing that you can imagine that you can have a variation between different cells, how these things will be organized. Specifically, this variation occurs across different cell types. Why does that happen? If we want to think about these differences, it's very useful to come back to the picture of how different cell types actually occur, where they come from, right? And that happens during a process called cell differentiation, which is the process during which a stem cell, which is a cell able to have any fate possible, when a stem cell becomes specialized, that's the process of differentiation. So here, as shown in this diagram, a stem cell can become anything. During this process, a dedicated transcription program is being initialized, meaning telling the cell that, well, now you will be a neuron, so there will be genes that you no longer need, right? So if then the genes that are, for example, needed for muscle cell will not longer be accessible or ready to be activated at second notice. So these genes will be, we call it silenced, and they will be really packed away, kind of like winter clothes. So you go from this transition from stem cells, where you have all genes poised to be used at any moment. We call them also hyperactive transcriptionally. And then after differentiation, you have silenced big parts of the genome with the genes that are no longer needed. So we started to think about this transition as a beautiful perturbation to a even more beautiful polymer system, and that's the genome. So you are going from this system that is completely homogeneous and with a homogeneous polymer solution, and you are reorganizing during differentiation, where you are silencing these genes, which are actually packed away in those little islands, which we hypothesized will be of a different phase. So how did we test this experimentally? I will get to this. So these islands of these silent genes we refer to in biology as heterochromatin. So to test this experimentally, we built a system, a mouse system, where we use mouse embryonic stem cells, which have fluorescently labeled histones, so histone H2B. So specifically these little pearls that I showed you, we have there fluorescent markers that allow us to visualize the genome, its motions, and so on, and we can follow its differentiation. We specifically differentiate to neurons. So we are making from mouse stem cells, we make mouse neurons, and what you are viewing here, that's only the nucleus, that's not the cell. You are looking here at stem cell nucleus, and once you differentiate, you see it becomes actually much larger, right? You have actually then neuron nucleus. And so what I want to point out here, what's visible kind of right away, is the density distribution spatially dramatically changes. So going from this homogeneous, right? So don't focus now on the holes here. Those are further embedded organelles. Focus on the fluorescent signal. So here the red you see is, there are some variation, but very small density variation in the stem cell, where in the neuron, we start to have these yellow islands of really high density, right? So you can also look here at the density distribution, and clearly there are these high density states that kick in after the differentiation, and that's exactly this heterochromatin. So now how do we test what are the physical properties, the material properties of this new phase that has emerged? So for that, we use displacement correlation spectroscopy that we have developed many years earlier, and has proven extremely useful to study nucleus-wide genomic motions in live cells. So with the DCS, we can measure here the genomic motions now in undifferentiated cells and differentiated. And from that, of course, we can extract a lot of information. So first, when you look at the local speed, this is the amplitude, right? So yellow again over here corresponds actually yellow to low speed. So the bluish regions are faster, yellow are slow. So you see that here compared to stem cells, which is like more or less bluish, the color differences are rather small. Here you have emergence of these yellow islands. Similarly, when we look at the local correlation of these displacements that we measure across the nucleus, we find that now for the color scheme is on purpose, yellow is high correlation, pink is uncorrelated. So again, you see stem cell is largely uncorrelated, the nucleus, the genome here, whereas in differentiated cell, you start to have within the pink uncorrelated matrix, you start to have the yellow islands of highly correlated genomic motions. And this, of course, so here to bring the full picture together, right, you can on first look already see the connection of these yellow islands across these different measurements. So what you will see is that as the density increases, so these yellow islands, the more dense ones, are the ones where we see the velocity go down, as well as we see the correlation here going up. Now, this is consistent with a picture that these yellow islands correspond to a solid-like gel phase. So you have these, as I mentioned, less mobile and correlated motions, whereas the surrounding, this pink or blue phase in these different representations corresponds to a liquid-like solid phase, since we have there, the motions show high mobility and actually are uncorrelated. But we can do even better. From this, we have here hypothesis that is already here supported with clearly showing evidence that there is a rheological phase separation with this sol and gel phase, and we want to go directly into these islands and see what are their rheological properties. So let me show you our spatially resolved measurements. So first, this is a mean square network displacement, which is an analogon of mean square displacement that we can obtain from displacement correlation spectroscopy. And each of the literally mean square displacement is a function of time. And this is for differentiated cell now average displacement across the entire nucleus. So you see it increases with time. So now what we are interested, how different will heterochromatin, so now these yellow islands, how different will be these displacements within this gel phase that we presume it should be. So we measure now displacements only inside, and when you look at it first, it's not very impressive. It looks very much like an average. However, if you consider and you start to look closer, you will see that your island itself actually moves. It undergoes actually dramatic translation, and this is its trajectory. So when you look at its centroid motion of the island, it looks like that. So of course, to get the true measurements of the displacements inside of my yellow island, I have to correct for its translation. So once you do that, you realize that you have a really constrained motion within this yellow island, so within the gel phase. So this really behaves as a solid-like gel. And we can do the similar analysis here for euchromatin, so the liquid-like phase. And indeed, you see there the displacement, they just increase with time, so consistent with a liquid-like behavior. So you see that indeed we have here that the differentiated genome can be described as a biphasic material, where you have the liquid-like matrix with these heterochromatin solid-like gel islands. Once we have performed this spatially resolved analysis, we wanted to do even better, and we're asking the following question. Well, if I can see these two phases so well, can I get their signatures also out of bulk analysis, right? Are they so strong that actually if I perform bulk rheology measurement, will I see the signatures of those two phases? So for that, we do following trick here. So we measure the mean square network displacements for both. Here the red ones are the differentiated cells, blue are the stem cells for their genome, and you see that there is some difference. So we want to see if the difference comes from the merging of the second phase in the differentiated case. So to go from these displacements to rheology, we take inspiration from the passive microreology. We use here generalized Stokes-Einstein equation to translate this into, to extract the viscoelastic moduli, loss modulus, and elastic modulus. However, for those of you here, active meta scientists in the audience, I want to point out that, of course, we're working here with an active system, and we have other evidence why here we are allowed to assume T to be actually for the given time scales frequency independent. So I'm not showing it here directly. Therefore, you see I'm actually here normalizing out the temperature as well as the size of the particle. So we're looking here at the functional shape of the viscoelastic modulus. That is what we're asking. We are not measuring absolute values. And what you can see here right away, the functional difference here, the shapes of the blue curves versus red curves, so our G prime and G double primes are different between stem cells and between the neurons. Now, is this difference coming due to the fact that they are actually one is one-phase material and the other is biphasic material? So let's do the following analysis. So first, when you look at the G prime and G double prime of undifferentiated cells, what you will see that the loss tangent behaves as omega minus 1 with frequency, which is a signature of a Maxwell fluid. Now, indeed, if we fit this model to our data, we get a perfect fit. So this is really impressive that the genome, the undifferentiated genome in stem cells indeed behaves as a Maxwell fluid with a single relaxation time. Now, what about the differentiated genome? There, I told you a second phase has emerged. So we know already here, this is the Maxwell fluid, this dotted line. Of course, it cannot agree with it. So how can we account, what's the minimum model to account for the presence of the solid phase? So for that, we add a minimum solid, which is a Kelvin solid, and this serial arrangement of Maxwell fluid and Kelvin solid gives us Berger's material. So when you fit this model to our data, we get a perfect fit. And what is really cool about it, this allows us now to extract actually the material parameters, so the Young moduli and viscosities of these two different phases, as well as the relaxation times. So this is here for kind of the summary of the first part of my talk that I wanted to show you, that indeed we found that the undifferentiated chromatin in stem cells behaves as sol, so as a liquid-like phase. In differentiated chromatin, you have a coexistence of two phases, sol and gel, and these new phases emerges during this formation of the heterochromatin. So it's the heterochromatin, the silence genes that correspond to this gel. So once we had this experimental evidence, the next question that came up was, how do we think about the physical origins behind this genomic reorganization, right? So what forces, what are the physical principles behind this large-scale reorganization? And to address that, we used large-scale computational models together in collaboration with Mike Shelley at Courant at NYU and David Santian at UCSD. And to describe the system, we developed a model where now we have, we are modeling the entire genome inside of cell nucleus. And let's first let me give you the physics of it, the main ingredients. So our polymers now actually consist of parts that correspond to euchromatin and parts that correspond to heterochromatin. What does it mean? I have euchromatic part, the orange ones, those are the ones that are where the genes are actively transcribed. So I have active forces stochastically acting on those, right? Then I have, however, the blue parts, that would be the heterochromatin. So those are the silence genes. So there are no active forces present there. Moreover, as this heterochromatic parts actually have the ability to cross-link and actually form this gel phase. In addition, we have also added tethers of the heterochromatin to the possibility of forming tethers of heterochromatin with the nuclear envelope as it occurs in vivo. Now, this entire system is, as I said, is inside of the nucleus. Here I'm drawing a sphere, but we went even further. We even model ellipsoid to mimic the closer shape of the real cell nucleus, and it's immersed in a fluid. It's immersed here in a viscous fluid to simulate the presence of the nucleoplasm. So now you can imagine as we let the system move, and you have, in addition to thermal fluctuations, you will have these active forces working. These active forces will lead to formation of fluid flows, which I will show right in a moment. So this is kind of what's under the hood. What the model here is basically beads connected here. Actually, this is a picture from our earlier model where we had rigid springs, rigid links. In this model, what I'm showing you here, these actually are here elastic springs in between the beads. But the idea is here that basically for every single bead, we're solving the Langevin equation, where basically for every bead, you consider the active flows that it encounters and the forces on it, such as Brownian excluded volume and the tensile force from the links. And then we solve the Navier-Stokes equation, which is here for the viscous flows, the Stokes equation form. So this is what you are solving then across the space. Now, so what will it show us? So as I told you, we put in the entire genome. So you have here, well, haploid. So you have here 23 chromosomes. They, you initialize, you let them run, and you see now what the hydrodynamic interactions on the top of all of these conditions that I explained to you, what they lead to. So the dynamics is rather complex. So let's look in more detail. So we can look now and analyze obviously the system under various conditions. How much time I have, Lulu?
8 minutes before questions.
Good, wonderful, perfect. So what we can now learn about, we can look separately how is our polymer behaving. We can look at the role of the cross-links. We can look at the formation of the heterochromatin, so how actually this solid-like gel phase, how this emerges, right? And here, as an example, we are picking 4 chromosomes that we are plotting here, kind of outside of this crowded environment, so you kind of get an intuition also how these polymers here behave. And so you see the conditions, well, for example, specifically that I'm presenting, is this row well, this is T0, but this row corresponds to a passive system, so I have no active forces. Here, this row is with activity without voltators, while the last row is the system that has activity with voltators. And you see that, for example, the presence of voltators is very critical. You prevent large-scale mixing by actually introducing these little staples of heterochromatin to the envelope. So this, you see, this is a very important for the genomic organization. And here, this is difficult from these plots to see, but I'll discuss in the next slides in detail. What can we see? What can we learn about the formation of the gel phase of the heterochromatin? One thing that I'm here, I kind of have not explained, but you can see that I'm listing here only extensile forces, right? So as on a previous slide here, which doesn't work, as you see, our forces here that they are applied in forms of force dipoles. And in our earlier work, which I didn't have time here to explain, we have already tested the effect of extensile versus contractile forces and found that for these large-scale organization, indeed, it's actually extensile forces for the genome that play the role. So that's the reason why we are focusing here in this specific work on the extensile. So let me show you here the role of the activity on the formation of heterochromatin. So first we can evaluate the chromatin density as a function of time. So you see here for the, if you let our system evolve in time without presence of active forces, you will generate regions of higher density simply due to the nature of the fact that we added that we allowed for the heterochromatin parts to form crosslinks, right? So they will actually bunch together and you will form these regions of higher density. However, note here, once you add activity, they will be boxed together much stronger, and the density actually of these heterochromatin regions is much higher, right? So this is first what we see is that the heterochromatin density indeed is enhanced by the euchromatin activity. Another thing that you can look at, we were very interested in the alignment of the fiber. Will we find any order across the nucleus for the fiber? So we have been measuring the local nematic alignment, and you see in the passive system there is no alignment whatsoever. However, once you add the activity, what you start to see that there starts to be alignment in the parts where you have the active parts of the polymer, the heterochromatic, the dense part that was already boxed there in the middle, that part of that does not have alignment. So you see that indeed now the euchromatin activity induces these local nematic orders in the periphery of the nucleus. And even more, we can go and analyze the local stresses in different directions inside of nucleus. So we can here look at the stresses on chromatin, both the radial component and the shear component of our stresses in the system. And so you see the distribution here in the passive case. So there is some heterogeneity to it, but it's not very strong. But once you add the activity, right, the differences, the contrast is actually, it's striking. So you see that there are large tensile stresses inside of the heterochromatin, and there are these active stresses, right, since we have our activity here localizes here, that's where the euchromatin, the phase separation basically of the active and inactive parts went. So those parts of the active parts of polymers are localized here. So this is where the amplitude is the highest. So indeed, this is another piece of evidence, right, that the euchromatin activity now enhances heterochromatin segregation. Now, the system, in our description, we include the presence of the fluid. In fact, it's one of the critical components. So it's the hydrodynamic coupling between the active forces that leads to a lot of this phenomenology. So we started to look also at the nucleoplasmic flows. So how is actually the fluid behaving in this whole question now? And what you can see, so these movies have stopped. These are flows here. These flows are due to euchromatin forces. These are flows due to heterochromatin. We call it forces, but what it is, it's this elastic recoil as the heterochromatin is kind of being pushed and tested there by the flows coming from outside from the euchromatin. And here you have plotted the total flows. And what we see, what we find here from this analysis is that it's indeed, once you have euchromatic activity present, you start to induce actually large-scale nucleoplasmic flows. So you start to have the flows that are at the scale basically of your system. So you start to have a large flows across the nucleus. Why this is very interesting for us, because there's earlier work from our group that I have not discussed today, where we had a discovery about 10 years ago that there are large-scale coherent motions, within the genome and within nucleoplasm. We found that there are large-scale motions that you observe in this genomic system. So that's just of note why this is here actually so much of interest. So we see that it's nucleoplasm actually here that plays a key role. Finally, one last piece of analysis that I would like to show you is how do we connect this picture, this physical framework that we have evolved, to the knowledge that comes out of the chromosome conformation capture techniques. Maybe some of you have heard, this is a standard way in genomic studies over past 10, 15 years by now, is to perform this biochemical assay of chromosome conformation capture that allows you to measure contact probabilities of different sequences within the genome. Long story short, let me explain here, what does it mean? If this is the sequence of my 1D linear sequence of my chromosome 1, and here behind it I just added chromosome 4, and I plotted here again against chromosome 1 and chromosome 4. So what I'm plotting here, the color, of course, the intensity corresponds to the contact probability that this guy and this guy are physically found in space together. And now how this plot is here generated, the bottom part, the red plot, is just a snapshot at one single time point that we take and go from our model, and we compute the contact probabilities. And the upper triangle here, that's the average over our entire measurement. So what you can see here along the sequence, I have here highlighted in blue, those are the heterochromatin blocks. So those are by definition in our model, right, defined as pieces of chromatin without active forces, but with crosslinks. And so those then along the diagonal, they basically are translated here into these green pieces. So now that we know the metrics of it, let's compare the passive case and the active case. So you see that clearly on a first look, these patterns look very different. And I will draw your attention only to a couple of details. So we start to see this very unique different pieces here. So we're focusing here on a position along the diagonal, so where we have the highest contact probability. And so we notice presence of these loops that are physically here presence of, this is my purple inset. You can see this is the confirmation of, one of the confirmations during the, here, time average. So here this is the snapshot. So from this snapshot, this is the polymer confirmation corresponding to that. Here you have from this teal inset that is here, this situation. So you see that we find actually formation of loops, and this is something very, very curious and interesting, because those are features found in the real genome. And the loops in the genome, that is currently one of the big questions. What is their role in the organization of the genome? These loops can also interact. They form actually at various length scales. So ours here, I have to stress here, ours are actually quite large. So we do not think that these are the loops that correspond to, for those of you familiar, formed by condensing and cohesin. So those are the structure maintenance complexes of chromatin. These are actually large scale loops that transiently exist and then will actually dissipate and reform over time somewhere else. So definitely so we see the presence only in case of the presence of loops is there only in case of active forces present in the system as well. And you see that the presence of activity also gives you this much more pronounced generation of these high density or highly segregated heterochromatic regions. So this is just another way to measure how strongly the euchromatic activity basically helps us to bunch up together the heterochromatin. So with that, this is my last data slide. So to conclude, I hope that I was able to show you here our novel methodology that we found to extract the rheology from live cells. I would like to stress that the way how we extracted the material properties is completely non-invasive, meaning we have purely looked at the cells, right? They don't even know we're there. We have not inserted any probes or anything, and we obtained these material properties. We find that the genome is indeed organized by this rheological phase separation, where you are generating a new emergent phase upon cell differentiation. I have also showed you from our hydrodynamic model how actually the presence of activity helps to generate this highly dense gel phase. And why do we care actually about all of these things? Why are we so excited to understand the material properties and the rheology spatially resolved in live cells? Well, this is extremely important because the nuclear material properties effectively impact all cellular processes via the central dogma, right? So it dictates then the time scales, how fast things can happen in the nucleus that will actually affect everything later downstream for all the other cellular processes. So with that, I would like to thank my team. Nothing would have happened without them. I have been very fortunate to nucleate an enthusiastic team of chromatin lovers at NYU. It has been a lot of fun in the Center for Submarine Research, so we've enjoyed ourselves a lot. I would like to thank, obviously, my collaborators, everyone in my lab, past and present members, our funding sources, and of course you for your attention, and I'm happy to answer questions.